So the innate immune system has to deal with things immediately. All right, so what we're going to look at today is what we call the immediate response to infection. As a reminder, our first physical defense against pathogens infecting us really are just the physical barriers of our body, membranes, whether you're talking about the tough outer skin or the lining of different types of tracts like our respiratory tract, GI tract, or reproductive tract. Right? I last time emphasized to you, this is a really important concept of understanding inside versus outside, right? Because even stuff that's going through your respiratory tract into your lungs or food going down through your GI tract are still technically outside of your body in the same way that a car driving through a tunnel, like the polytunnels, right, is passing through the mountain, but is not actually in the mountain, right? And that's a very important distinction. Because our definition of infection kind of only makes sense when you realize that it just has to do with when pathogens somehow cross through those barriers and actually get into our underlying tissues. So let's look at what happens here. Imagine you get a cut on your skin. Right? That moment you get a cut is the moment infection technically starts, right? The moment bacteria on the outside of your skin or on something like the pin or whatever that pokes you actually breaches that protective physical membrane of your epidermis and gets into the underlying tissue, that is the moment infection starts. Fortunately, underneath your outer skin layer in your dermis, you have innate effector cells already patrolling, right? MICR is there. Right, in plentiful amounts. And so, for example, macrophages, right? Macrophages are our MICR. They are already patrolling in the skin. And so the immediate response is macrophages that happen to be right there, right, come in contact with the bacteria that's coming in. So these rod-shaped, hot dog-shaped bacteria come in. These macrophages then go to work, phagocytosing or eating them and destroying them, right? At the same time, certain receptors on the macrophage are picking up on maybe different types of products of the bacteria that are being released. Right? So that combination of eating the bacteria as well as maybe just picking up on the presence of bacterial signals that are floating around the macrophage, right, trigger the macrophage to go ahead and release little protein messages. These little triangles, right, are small proteins that we call cytokines, right? And the function of the cytokines then is to alert the rest of the immune system that something's going on, right? Like the analogy of Mall Cop, I said that's like Mall Cop who's trained to recognize rainbow hair on the clown, right? And when he sees that, he gets on the walkie-talkie and he says, "Hey, I see a clown here. It's got rainbow hair, and that's what I'm trained to recognize." Right? So that message is the cytokine message going on, and it spreads out. So what are these cytokines doing? Cytokines function to alert the immune system and cause things like inflammation to happen. We will learn about the importance of inflammation and why it's such an important part of our immune response. Right? And importantly, these cytokine signals can attract other immune cells to come and help out. So the recruitment of other monocytes that are floating through the blood, so that those monocytes know that something is going on in the tissue here surrounding the blood vessel, so that they'll squeeze out through the walls of the capillary, right? So this is a capillary in cross-section. This is the very thin wall of the capillary. The white space would be the lumen of the capillary in which all the blood is flowing, right? So these monocytes would be recruited by cytokines. They'd squeeze into the solid tissue, and they would finish becoming full-fledged macrophages. Right? Other types of innate immune effector cells like neutrophils would also be recruited, and they would also help contribute. Right. So therefore, there's two sides or two, let's call it stages of our innate immune system. Right? The immediate response are the macrophages and other proteins that happen to be right there, right, to detect immediately and maybe start destroying the bacteria. And then there's the induced response, right, where all the cytokine messages that are released by the macrophages begin to attract or recruit other immune components to help out. Most of the time then, right, this sort of 1-2 punch of your innate immune response is all that you need. Right? So maybe within a few hours, the bacteria have all been eaten up. During that time, your wound is already beginning to kind of heal up, right, and sort of scab over. Right? And so then that's the end of infection. And you didn't even know it, right? You didn't even know you were infected, and your innate system took care of it. So let's look at this a little closer. So macrophages engulf bacteria via phagocytosis. So this is a really cool image on the left, right? This is an electron micrograph image of a macrophage, this big red cell here. And you can see it is attacking these bacteria, right? And so it is reaching out and grabbing the bacteria, and it's pulling the bacteria in. And you can see these bacteria that are now pressed up against the outside of the macrophage, they're about to get enveloped or taken in. So look at this cartoon here where we can see that, right? So the bacteria get brought to the macrophage, and the macrophage takes it in these what we call phagosomes. Right? And then these vesicles that contain the bacteria end up fusing with other vesicles that contain all kinds of enzymes that would essentially kill and digest and break down the bacteria, right? Thereby neutralizing it. Right? So this poses some interesting kind of challenges. There's only a limited number of bacteria that will happen to be sort of encountered by a macrophage in your skin, right? Or under your skin. It would be much more efficient if the macrophages had a way to really be sort of attracted to the bacteria and grab hold of the bacteria, right? Because the more that the bacteria can be held onto, then the more bacteria a given macrophage can eat, right? So how does the macrophage know where to reach? How does the macrophage know what to grab? Right? Some interesting questions here, right? I've always loved macrophages. I think macrophages are your like quintessential cliche immune cell, right? Because anytime you see an immune cell pictured, a white blood cell, right? Because remember, macrophages are leukocytes, but there's several different types of leukocytes. But generically, when we see a white blood cell, it's usually this big blob of something that eats something, right? And that's the macrophage. When I was a little boy, I think when I was like maybe like 10 years old or something like that, I saw this movie on TV. And this was an old movie that was actually made, I think, in the 60s or something. And it was called Fantastic Voyage. And the premise of this movie was, I think it was like the president of the United States or something like that had some kind of like, I don't know, maybe inoperable tumor or something like that. And he was going to die. And so the only way that the doctors figured they could treat him is somehow if they can get in to his body and actually battle the tumor there. All right. And so the way they did this was they got a team of surgeons and they zapped them with this special shrinking ray, all right, that shrunk them down, teeny tiny microscopic, and they shrunk down this kind of like submarine thing for them to like then be in, and then that got injected into the president's body. And the whole movie was about them then going around inside the body and encountering stuff. This is total 60s, right? So that whole idea of like submarines and things was kind of big. But anyhow, so there's this one scene where a big macrophage comes along and it starts attacking the vessel. And on the top of this vessel, there was this like little glass dome where there was like a seat inside. And one of the surgeons is sort of seat belted into this because that's like the observation thing. So this macrophage comes along, right? He gets stuck because his seatbelt won't come undone. And so his fellow crewmates are frantically trying to rescue him, but they can't, right? And then this macrophage ends up like busting through the glass and like eating him, right? And so when I was a kid, man, this was like the most horrifying thing I've ever seen before. But it was awesome, right? So then later, I had my little Star Wars action figures like Luke Skywalker, and I had these like styrofoam, packing peanuts, and I'm like mashing them into the Luke Skywalker. And my mom's just looking at me like, what are you doing? Right? And what are you doing to your toy? And I'm like, Mom, these are macrophages, right? And then I think she's like looked at my dad and she was like, that's your son, right? But anyhow, so macrophages are the quintessential right? First line of defense in terms of cells that attack things, get into your body, right? And they do so in a wonderfully dramatic manner. Right? But let's think about this. Right? Bacteria have evolved to be very clever in how they can avoid being detected by things like macrophages. The outer coats of many bacteria, especially gram-negative bacteria, have very thick polysaccharide coats on them. So here's an E. coli, right? And if you zoomed in on its surface, what you would see is you have this cell membrane down here, then you have this peptidoglycan layer, and then you have an outer membrane embedded in which are all of these long polysaccharide chains, right? Each one of these little hexagons, for example, right, is an individual sugar molecule, right? And they are attached to these lipids in the membrane. So we call these types of molecules lipopolysaccharides, or LPS. These lipopolysaccharides basically serve as a very thick shag carpet, right? Because three-dimensional features such as proteins and things like that are part of the outer membrane of the E. coli, which could serve as unique three-dimensional shapes that immune cells of your body might be able to pick up on and detect, those shapes are hidden underneath this LPS layer, right? And so this shag carpeting makes it very hard for things like macrophages to be able to properly attach and grab onto the bacteria. So how do you overcome this challenge? Right? If you have these slippery bacteria, right, with these thick lipopolysaccharide coats, right? How do you have any chance of seeing individual unique proteins inside of them for your immune system to be able to recognize? Well, one thing you can do is you can attach easy to recognize marks to these lipopolysaccharides, right? Sort of marks that your cells have special receptors to easily recognize, and therefore you can make these slippery bacteria much stickier. So look, here we go. Here's an example. Here I have a beach ball, right? Well, that's a beach ball, but it's actually not a beach ball. It's a very physiologically correct model of a macrophage, right? No, it's not. Okay. And here we have ping pong ball, right? So this is a slippery bacteria, right? It has this slippery plastic coat, and the bacteria. Oh, this is, by the way, this is a nice example because size-wise, this is pretty much to scale, right? If this is the size of a bacteria, this would be the size of one of your macrophages, right? But the ping pong ball is really slippery, right? It doesn't attach very well, and it kind of bounces off. Sorry, I lost it. So how can we make that slippery ping pong ball, right, stick to this macrophage? Well, we can do it through the help of a very particular special group of proteins, right? And these proteins, off here, right, you can stick them onto the ping pong balls, right? And now these ping pong balls can be more easily detected. So these proteins is a special family of proteins called Velcro. No, it's not called Velcro, right? These are called complement proteins. And so once you coat the surface of a pathogen with complement proteins, right, they can now stick much easier to the macrophage, right? And the macrophage has a much easier time grabbing onto and more efficiently eating more bacteria. So this requires that what? This requires that the macrophage itself has particular receptors on its surface, right? So in this case, it's the sort of fuzzy felt part of the Velcro. This part that got stuck on the ball is the little teeny microscopic plastic hooks or whatever, right? And so that allows you then to detect the complement proteins, right? So complement proteins coat the pathogen target, and then on the macrophage's surface, right, there would be special complement protein receptors to be able to make it so that the macrophage can then have an easier time binding to the pathogen target, right? Okay. So complement proteins tag pathogens for destruction. Right? So these complement proteins are a system of different soluble proteins that are found in your blood plasma. They're made by your liver. Your liver makes complement proteins, and then your blood is coursing, filled with these complement proteins, right, that then get into your tissues, and they're ready to attach to things like pathogens. So the modes of action of these complement proteins are that they make phagocytosis much more efficient, right, so that the macrophage can eat more bacteria. Also, complement proteins can serve as little signals that attract macrophages. So you can imagine in your tissue where you have a lot of complement protein activity going on, that's probably because there's lots of complement that's coating bacteria, right? So that can serve as a beacon or a sign to attract more macrophages there. And as we will learn, just the coating of complement onto something like a pathogen might already contribute to the disruption of that microbe, right? Leading to, for example, the destruction of the bacteria before something like a macrophage even necessarily has to come along and eat it up. So complement is a system of proteins then, right? It's not just one complement protein. There are many. In fact, there's probably about 30 different types of proteins that are all considered complement proteins. But the most important one is complement component 3, right? Or complement C3, right? So here's an illustration from the book. This blue egg-shaped thing is this sort of super simplified geometric sort of representation of a complement protein C3. Right? There's 3 basic steps as to what happens here. This complement C3 will be floating around in the fluids of your tissue. When it actually comes across something like a pathogen, right, it will be cleaved. So part of it will be cut off. So the C3 then becomes 2 parts, a small part that floats away that we call C3A, and the other part that's left that's called C3B, right? So part 2 then is this C3B is what's going to end up tagging the pathogen. Right? And the part 3 is this will now serve as that easily recognized signal that something like a macrophage that has complement receptor on its surface can now bind to, thereby making this otherwise slippery bacteria suddenly much more attractive and easily bound to the macrophage. This is also the first time I'm going to warn you about the illustrations in the book, okay? Because many of the illustrations of the book are super simplified like this because they're trying to emphasize the concept, but there's a danger in that, right? Because they often overlook or distort things that can be misleading. Number 1, for example, complement C3 is not really a blue egg, right? I think you guys know that. It is a folded up protein. Right? And number 2, the size scale that's shown here. I mean, this makes it look like one complement C3b is like just as big or even bigger than one bacteria, right? And that's not true at all. Because remember, the C3b itself is just a teeny tiny small protein, right? And the whole outer surface of the bacteria is made of like a bazillion small little proteins, right? So this thing attached to the bacteria, you wouldn't even see it at all if this was drawn to scale. Right? But the point is, if the whole surface of the bacterium becomes covered with these individual C3bs and you have a whole lawn or a whole coating of C3b on there, then it is just like the Velcro, right? One individual little plastic hook in the Velcro isn't going to do much. But if you have a whole bunch of them, right, and then you have the other fuzzy felt part of the Velcro pressed up against it, then you get very strong attachment. Right. So how does this C3b part of the complement protein attach to the surface? I mean, what activates the C3 in the first place, right? And how does the C3a part that floats away attract macrophages? Okay. So the complement protein attaches to the surface of a pathogen target using a thioester bond, right? And you're like, oh no, it's OChem. OChem rears its ugly head yet again, right? Yeah, you totally thought that you were done with OChem, right, in your entire life. But no, okay, we do need to talk about a little OChem here, right? So here's a thioester bond, right? So remember, an ester bond is if you have a carbon double bond to an oxygen, and then you have two different R groups attached to it. So thioester is just a little twist on this, right? Because thio refers to sulfur, so you have a sulfur group in there. Okay. Thioester bonds like this are highly reactive bonds. They love forming covalent bonds with whatever sort of they can, right? So here is the simplified blue egg representation of a complement C3, right? And you'll see that tucked inside of it is this thioester bond, right? Down below is a much more realistic view of what this complement C3 looks like, right? Because again, it is a protein, right? It is a single polypeptide chain. So I think down here, I don't know, maybe this is the end of the, oh no, sorry, over here, right? This is the very end, the very first amino acid of this long polypeptide chain. And if you were to play this little game and follow it around like a maze, right, you'd wind your way all around, all the way around, and eventually you'd reach the C-terminal end, right? The carboxy terminal end and the amine terminal end. So this is where the Bio 275 stuff comes really handy, because those of you who have taken Bio 275, hopefully, yeah, you're like, oh, I remember this kind of stuff, right? For those of you who haven't, and who are right now wide-eyed looking at this going like, I have no idea what any of this means, right? Maybe it's time to find a way to review some cell biology stuff and kind of catch up here, right? I did give you fair warning that Bio 275 is important for this class, okay? Anyhow, so you'll notice that the thioester bond is buried deep within this, right? And it actually comes from the fact that you'll have like two cysteine residues in here that have sulfurs as part of their side chains, right? And therefore they, two cysteines that are next to each other, right? They may interact in such a way to create kind of this little thioester form, right? So the C3 gets cleaved, so part of it falls off, right? And that small part is the C3A that floats away. So that is this part here, this red part here, which is labeled ANA or anaflatoxin. Right? When that floats off, what happens to the rest of the protein? Right? The whole rest of the protein shape has to change to kind of adjust for the fact that part of it has been removed. Right? And as it rearranges, what happens is the part of it that has that thioester bond ends up pointing down, right, over here. And this serves as a nucleophile. Right? And so it undergoes nucleophilic attack on the thioester bond. Right? Because this is floating around in water, you can imagine that almost immediately water interacts with the thioester bond, right, turning it into just a sulfhydryl group and the COOH. And guess what? If that happens, then this C3b is inactive. It's just going to float away and not be useful anymore. Right? But if this cleavage of the C3 happens really close to the surface of a pathogen, there is a chance that the thioester bond will actually form its covalent bond with some molecule that's on the surface of the pathogen, like, for example, one of those lipopolysaccharides, right, that are sticking out. Okay. So there are three pathways that lead to complement C3 being activated. I'm just going to introduce them today. We'll look at these in more depth over the next couple lectures. Right? They're called the alternative pathway, the lectin pathway, and the classical pathway. These all have to do with how different mechanisms take place to eventually lead to the activation or the scissors cutting the complement C3 so that the C3 can now bind to a target. Right? Today, we're going to start off looking at the alternative pathway. And this has to do with the fact that the pathogen surface, right, really close to it, is a good environment that favors complement C3 to start activating. Right? So the closer the C3 is to the surface of the pathogen, right, the more likely it is it'll activate, and therefore more likely that the C3b will get attached. And this alternative pathway is the first to act. All right. So I'm going to take you through some pretty horrible looking pathways here. Unfortunately, these are some of the more trickier pathways to understand in terms of these protein pathways. And unfortunately, it's at the beginning of the semester. But again, that's just because this is all part of the innate system. So we just have to get through it. But bear with me, and I think you'll be okay. Right? So step one here is here you have the surface of a pathogen. Here you have a complement C3 that's floating around in the fluid. surrounding the pathogen. It gets really close to the surface, right? And what happens is that the C3, right? So this is not C3b yet, right? Because it's floating in water near the surface, it will interact with water, right? And it becomes sort of ready to react. So we call this IC3. This is the first step, right? And the surface of the pathogen, right, is conducive to that because of like the pH and things like that, right? Step 2 is that this IC3 binds a different complement protein, complement B. That's this yellow thing over here. Okay? And then factor B acts as a zymogen, which basically means it's an enzyme that's going to carry out some function, but in its current shape or form, right, it's inactive. But you'll notice by binding to the complement C3 over here, right, the factor B changes its shape a little bit, and you get this little groove, right? And this little groove is where this next complement protein, factor D, comes in and binds to. Okay? That factor D will bind and it will cleave this yellow factor B, turning it into a BB and a little BA that floats away. So now this resulting complex we call IC3BB. Okay? So what this IC3BB does now is this BB is now active in terms of its enzymatic activity. And what it likes to do is it likes to cleave other C3 proteins. So another C3 protein floats along over here, right? Attaches to the IC3BB, and the yellow part, the BB part, will cleave then this C3 that came along, right, releasing a C3A. And since this is taking place really close to the surface of the pathogen, the C3B, its thioester bond is exposed and it gets planted there. Right? So that was step 4. Right? So because this complex in step 4 converts C3 into C3b, we call it a C3 convertase complex. Okay. So the next slide continues from this point on. But we'll start with this C3b that's already on the surface, and we're going to move it all the way to the left, right? And here it is. So this process happens again, the exact same process involving factor B, factor D, turning it into a convertase complex, and it cleaving another C3 that floats by, planting that C3b on the surface, right? And once it does that, this convertase complex, after it's given up that newly cleaved C3b, right, it's free to keep going around and around and around, grabbing another C3 and making another C3b. Importantly, each one of these C3bs can also turn into a convertase complex. So once this starts going, very rapidly, you would get this spread or coating of the surface of the bacteria with C3bs, right? You are now putting the Velcro on the surface. Right? And this convertase complex, because it's attached to the surface of the cell, we call this the alternative C3 convertase. Right? So if this is just like the slide before it, what's the difference? The significance is that now you are starting with something that's already on the surface of the cell rather than a C3 that was floating around somewhere else. So therefore, once you get this process started, right, it becomes way more efficient to keep attaching things directly to the surface of the cell. Right? So that's very important to remember. All right. This coating of the surface is what we call opsonization. When you coat the surface of a pathogen target to make it easier to detect, right, you say you are opsonizing that pathogen. Right? So how do we control this? Because obviously this looks like a chain reaction, right? There are certain regulatory proteins that control the deposition of C3b. There are certain factors like this pink protein, properdin or factor B, that stabilize the proper shape of a C3 convertase, thereby making it more efficient for it to carry out its job. So this would increase the opsonization rate of the bacteria. Other factors like factor B or factor I work by cleaving or destroying the C3b that's on the surface, right? So that would slow down the opsonization of the cells. Keep in mind that the thioester bond from the C3 complement, it doesn't discriminate between a pathogen versus like a normal cell in your body, right? So the normal cells of your body might accidentally start getting opsonized, and that would be bad, right? So how do we prevent that? Well, we have certain proteins that are made by our own cells to discourage them from being opsonized. So decay accelerating factor, you can see here, this purple thing removes the yellow BB part from the C3b, right? That would slow things down. Or this membrane cofactor protein, right, over here, right, actually attracts factor I to come and cleave and break up the C3b. Again, slowing things down so that your own healthy cells of your body don't get improperly opsonized and targeted for destruction. Right. This is the last slide for today because I know I'm going over time. So as I explained, right, the process of opsonizing with complement protein greatly increases phagocyte efficiency. So here we have a macrophage. Here we have a bacteria that's being coated with C3b. Again, the size scale is not correct, right? Because the reality would be they wouldn't see individual C3bs on this bacteria. But the point is, just like the Velcro has two parts, right? Here you've put, one part of the Velcro on here, and then on the macrophage are these complement receptor proteins or receptors, right? CR1 that does the actual recognition, bringing the bacteria in, making it stick a lot better so that the macrophage can more effectively destroy it. Right? So the effectiveness of opsonization through complement proteins is the reason that most pathogen invasions in our body are eliminated right from the start of infection. And within hours, something from a paper cut, right? You're no longer bothered by it because it's already healed up and your innate system has taken care of all the bacteria just through the effects of complement proteins and macrophages. Okay. All right. So that's it for today.